Okay, ladies and gentlemen, good day all. Today we are going to look at the concept of structure. This PowerPoint is not made by me, it's made by Wadakant from Kerala, India, from the University of Leicester. So, what is on this concept we are going to look at? Uh, we are going to achieve the following objectives. Number one, to understand the history of gene. Number two, to learn the structure of prokaryotic gene and to delineate on eukaryotic gene. And then lastly, to study silent features and characteristics of gene. So now let's start with the concept of DNA. What is a DNA? So a DNA is a double-stranded structure. It is a double-stranded structure that made up of 5' end to 3' end, which is the same strand, and then also 3' to 5' direction. So this is the structure of DNA. So it's made up of double-stranded structure. So this DNA is very, very important. In fact, we call it the language of life because all the activities of life of every living organism is controlled by its DNA. And of course, this DNA is located in the nucleus of the cell in the case of eukaryotic cell, while for the prokaryotic cells, it is located in the nucleoids. And this DNA is very, very important and essential. And the genes are actually found in the DNA. So the genes are the segment of the DNA that involve in the synthesis of protein. And now we are going to look at the definition of the gene. What is a gene? A gene is a specific sequence of DNA containing genetic information required to make a specific protein. So it means that if you have a DNA like this, if you have a DNA, so in a DNA, there are segments. There are different segments, and each of these segments that produce a functional proteins are called genes. So in one DNA, we can have 1,000 to 100 of genes. Like for example, this one can be a genes, let's say gene 1, this one gene 2, and gene 3, and this one is gene 4. So all these genes, they are like a segment in the DNA. They are like segment of the DNA, and each of the segments are responsible for producing a specific protein, and each of the specific proteins has its own biological activity. So let's say now this is gene 1. So in gene 1, generally from the DNA, gene 1 have the ability to transcribe to produce what you call messenger RNA, to produce messenger RNA. And this messenger RNA can further be translated into a functional protein, and each of these proteins have a biological activity. So it means that a gene is a segment of DNA. It's a segment of DNA that can produce a functional protein. And we have different types of gene based on the organisms. So we have prokaryotic gene. It means that there are genes that are found in the prokaryotic cell, and we have eukaryotic gene. And these eukaryotic genes are those genes that are found in eukaryotic organisms. So as I said, we have a concept called Central Dogma of molecular biology, where in a DNA like this, there are thousands of genes. And these genes have the ability to produce a different messenger RNA. So each gene can produce a specific messenger RNA, can produce a specific messenger RNA, and each of these messenger RNA can produce a functional protein. So the process whereby DNA is converted into RNA is called transcription. And then when mRNA is converted or is translated into protein, the process is called translation. And then each of these proteins have a specific biological activity. It has a specific biological activity. Like for example, we can have a gene for hemoglobin. We can have a gene for hemoglobin, HB. And of course, we know that hemoglobin is a protein that found in the red blood cells, which is responsible for transport of oxygen. So, as I said, a gene is a segment of DNA that can produce a functional protein, and each of these proteins is what make us the way we are. Like for example, we as a human, we have a gene called FOXP2, which actually make us to talk. So those organisms that doesn't have these types of gene, so each of these genes have the ability to produce a functional protein, and it's actually these proteins that make us who we are. Like in our mouth, we have what we call FOXP2, and FOXP2 is a gene that actually expresses the proteins that allow us to talk. So it's not every organism that talk because they doesn't have such genes. So genes is very, very important. In fact, it's what makes us the way we are, the color of our skin. If you are intelligent, if you are brilliant, if you are beautiful, it's all because of the different types of genes that you produce and also express in our body. So we have different types of genes. As I said, we have prokaryotic genes. Prokaryotic genes are those genes that are found in prokaryotic organisms. An example of prokaryotic organisms, we have bacteria and cyanobacteria. And then we also have eukaryotic genes. Eukaryotic genes are those genes that are found in higher organisms. And examples of these higher organisms, we have plants and animals. And how do you consider organisms as a higher organisms? Is those actually organisms that have the ability, are those organisms that have a nucleus and membrane-bound organisms. So all those organisms that have nucleus and membrane-bound organelles are called eukaryotic organisms. So therefore, eukaryotic genes are those genes that are found in eukaryotic organisms. And then the next types of gene is actually based on the activity. So genes are also classified based on their activity. We have what we call housekeeping genes. So what is housekeeping genes? Housekeeping genes are those genes that are always active. They are always expressed. They are always produced in your body. An example of these housekeeping genes are those genes that are involved in glycolysis. Because almost every time glycolysis are taking place in our body. So all the genes that are involved in glycolysis are examples of housekeeping genes. The genes that also allow us or that make us to talk, because usually we talk, the genes that are used actually in seeing, like opsin and crystalline proteins, are all examples of housekeeping genes, because we always use those proteins to see. So all those genes are example of housekeeping genes, are the genes that we produce them all the time, and they are always active in our body system. And then the next thing, apart from housekeeping genes, we also have specific genes. So specific genes are those genes that are produced when they are only needed. They are the genes that are produced when they are only needed. And of course, we have such kinds of genes in our body, so we call them specific genes. We only express them, we only produce them when our body is in need of them. And then of course, housekeeping genes are those genes that we need them all the time. So these are the types of genes according to their activity. And then the next thing, another types of genes. So we have another types of genes based on behavior. So another types of genes based on their behavior. We have basic genes. So basic genes are the fundamental genes that bring about expression of particular character. So they are the genes that we have them in our body. Their main functions their main functions is actually to bring or to cause the expression of a particular character in a body system. So those kinds of genes are called basic genes, the cause or the bring about the expressions of a particular character. So once they are there, they will influence some characters to be produced. So those kinds of genes are called basic genes. And then we have lethal genes. So lethal genes are those genes that bring about the deaths of their processor. So it means that any organisms or any cells or anything that have lethal genes, it means that that cell will automatically end off. It will die. So that is the functions of lethal genes. They are the genes that you don't need them. Sorry, that actually whenever a cell have such genes, then that cells will automatically go, it will die. Then another type of genes is also a multiple gene. So when two or more pairs of independent genes act together to produce a single phenotypic trait, that type of character is called that type of genes is called multiple gene. So these are actually the kind of genes that violates the law of Gregor Mendel. So an example of those characters that are influenced or that can actually produce a single character, two or more of them that produce a single character, is the ABO blood group system, that is blood grouping. So it's an example of multiple genes. So it's when you have two or more pairs of independent genes, they are independent of one another, but as a result of bringing themselves together, they will actually lead to the production of a single phenotypic trait. And then the next thing after multiple gene is cumulative gene. Cumulative genes, it means that are the genes that have additive effect. They have additive effects. So when they come up together, when they sum themselves together, so they actually influence the action of other genes. So this is what we call cumulative genes. So some genes have additive effects on the effect of other genes. So they will sum themselves, they will bring themselves together, they will add themselves together, and then they cause the actions of other genes. So these are called cumulative genes. And then the next thing after cumulative gene is pleiotropic genes. So pleiotropic genes are the genes which produce changes in more than one character. So they produce changes in more than one character. So those genes are called pleiotropic genes. They have the chances, they have the capacity and the potential to change more than one character. And then we have modifying gene. What are modifying gene? Modifying gene are those genes which on themselves cannot produce a character by itself, but they interact with other to produce a modified effect. So these kinds of genes are called modifier gene. On themselves, they will not express any character. Rather, they interact with the other genes to modify the effect of that particular gene. So these kinds of genes are called modifier gene. Like for example, now assuming now you are someone that you cannot talk, and the problem is from your gene. So if you can have a gene that will modify that gene and make you to talk, so those kinds of genes are called modifier gene. And then we have inhibitory gene. So what are inhibitory genes? So these are the genes that usually what they do, they suppress the expression of a particular gene. They suppress the expression of a particular gene. Assuming now you have a particular disease which actually came or which actually happened as a result of a production of a particular proteins in your body. So I think in a simple way, you can just get this kind of inhibitory gene that can just directly go and inhibit those genes that are responsible for producing those proteins or that protein that caused the disease in your body system. So you can use these inhibitory proteins to suppress or to inhibit the expressions of that particular gene so that to stop the expression or the production of that proteins. So this is what you call inhibitory gene. So these are the types of genes based on their behavior: basic genes, lethal genes, multiple gene, cumulative gene, and we have pleiotropic gene, and then we also have modifying gene, and then lastly inhibitory gene. So now the next thing is the history, introduction and history of gene. So the classical principle of genetics were deduced by Gregor Mendel in 1865 on the basics of breeding experiment with peas. So he assumed that each trait is determined by a pair of inherited factors, which are now called gene. So actually, when Gregor Mendel carried out his experiment on genetics, he used a breeding experiment where he bring about a piece of different color, and he was able to produce offspring, some inherited the characters from both parents. And that is how he came up with an idea of dominant character and recessive character. So from there, from his experiment, he was able to actually came up with an idea that yes, the traits in which those offspring from the pea plants obtain is due to inherited factors. And that inherited factors is it was not known as a gene then until 1909 when Wilhelm Johansen coined the term gene. So at that time, Gregor Mendel don't know about the gene, but he knows that the character that passed from the parent to the offspring is due to inherited factor. And that inherited factors is coined by Wilhelm Johansen in 1909 called gene. So what is this gene? As I said, ladies and gentlemen, in a cell, we have what you call nucleus, and this is the nucleus. So in a cell, we have nucleus. So this is our cell, and we have nucleus, and in the nucleus, we have chromosome, and in the chromosomes, we have a gene. And this gene, in the chromosome, we have DNA, and in the DNA, we have genes, and this genes is responsible for producing a functional protein. So I think, ladies and gentlemen, in a simpler term, you can now understand that we have a cell, and inside a cell, we have nucleus. And inside the nucleus, of course, we have what you call chromosome. And this chromosomes number varies across species. And then, of course, in the chromosomes, we have DNA. And in this DNA, of course, we have thousands of genes. And then this gene is responsible for producing a particular functional protein. So this is what we call a central dogma of molecular biology. So we should understand that in eukaryotic cells, we have nucleus, and in the nucleus, we have chromosome, and in these chromosomes, we have DNA, and in the DNA, we have thousands of genes, and each of that genes is responsible for producing a specific functional protein. So then now we are going to look at the prokaryotic gene structure. So in the prokaryotic gene structure, it's made up of different components. We have what you call promoter region, we have terminator region, and then we have what you call RNA coding sequence, and then we have also transcription initiation region, and we have transcription termination region. So RNA coding region is a region where a functional protein can actually be obtained. And then promoter region is a region where a functional protein called RNA polymerase bind to and then initiates transcription. And then terminator region is a region where your transcription stop. Usually you then stop codons. And these stop codons include you are away, that is UAA, 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 you are away, you are gone, then you go away. So these are the three stop codons. These are the three stop codons. So whenever there is transcription taking place in a gene, and then the transcription or the RNA polymerase come in contact with any of these three codons, then the transcription will automatically stop. So that is why we call them stop codons. UAA, that is you are away, you are gone, you go away. So these ones we call them stop codons. We also have start codons. Start codons, that is AUG. AUG is a start codon. AUG. Are you gone? Are you gone? That is AUG, which code for an amino acid called methionine. Methionine. So this is the start codon, and it's usually start with methionine. So ladies and gentlemen, in prokaryotic gene, we have three different components, which is promoter, sites, RNA coding sequence, and then we have terminator sites. So the next thing is actually the component of this prokaryotic gene. As I explained, we have promoter region, we have RNA coding sequence, and we have terminator region. So prokaryotic gene is continuous and uninterested, where there is no intron spreader. So generally in a DNA, we have introns and exons region. So introns is a non-coding region. That is a part of the DNA that do not code for messenger RNA. And then exons is a region that code for messenger RNA. So generally speaking, prokaryotic cell doesn't have introns. They only have exons. That is the coding region. And then the 5' of the promoter sequence is called upstream sequence. So in the promoter region, we have, in the DNA, we have upstream sequence and downstream sequence. We have what you call upstream sequence and downstream sequence. Let's say this is our gene. So we have what you call upstream sequence. We have upstream sequence, and then we also have downstream sequence. So in the downstream sequence is where we have a terminator region, which is the 5' end. Sorry, the 3' end. The 3' end is the downstream sequence. And then the upstream sequence is 5' direction. So it means that in the 5' direction, we have a promoter sequence. And in the 3' direction, we have terminator sequence. So now we are going to look at the promoter gene sequence. So in the promoter gene structure is made up of -35 region, -10 region, then we have RNA start region, where the protein synthesis starts. So this is situated on upstream. So it's situated at upstream of the sequence that code for RNA, and this site usually interact. It is the sites that interact with RNA, which is the key enzymes that involved in transcription. So the RNA polymerase is the enzymes that usually bind, or that must bind to, the promoter region before transcription start. So, and this promoter region also provide the location and direction to initiate transcription. So transcription is initiated at this site, which is promoter region. And the promoter region is made up of -35 region, -10 region, and then RNA start site. So in the -35 region, we have TTGACA. And then we have also Tata AT, that is TATAAT. So these are the -10 region in the prokaryotic organisms. And then, of course, in the prokaryotic gene structure, we have enhancers and we have also activator. So at at -10, there is a sequence called TATAAT or Fibnobox. And at -35, we have another consensus sequence called TTGACA. So these two are the most important promoter element recognized by transcription factors. So this -35 and -10 site are the site where the transcription factors always recognize and bind to. The transcription factors are the smaller protein sequence that usually bind to the promoter site to enhance or to activate transcription. And then the next thing is, of course, the RNA coding sequence. So the RNA after promoter, then now we are going to look at the RNA coding sequence. So the DNA sequence that will bring or that will become copied into mRNA molecule, that is the RNA transcript. So RNA coding region or RNA coding sequence is a region or is a DNA sequence that usually copies into a specific RNA molecule. And it starts with an initiator codon. It starts with an initiator codons and ends with terminator codon. So the initiator codon is AUG. And then the terminator codons are those 3 stop codons that I mentioned earlier. Then there is no introns. So it is uninterrupted with introns. And it is co-linear. It is mRNA, that is messenger RNA. So any nucleotide sequence present on the left is donated by minus symbol. And the region called offspring. So it's called upstream element. So they are called upstream element, and they usually have a negative sign. And then any sequence at the right of the start, which is at the downstream element, they are numbered as positive. That is starting from +10, +35, etc. And then the next thing is terminator region. So the terminator region is a region that signals the RNA polymerase to stop from the DNA template so that at least the transcription to be terminated. So transcription termination occurs through a row-dependent or row-independent manner. So these are the 2 ways in which the transcription stops, either through RNA-dependent manner or RNA-independent manner. And then the next thing is eukaryotic gene. So in a eukaryotic gene are more complex than that of eukaryotic, and they are composed of the following regions. They have exons. Exons is a coding region. That is a region that can actually transcribe to produce messenger RNA. And introns is non-coding region and is usually found in eukaryotic gene. And then we have promoter sequences. There's also terminator sequences, upstream sequences, downstream sequences. There's also enhancers and silencers. And then we also have signals. So these signals, that is upstream sequence signal for addition of 5 cap, that is 5' cap. And then at the downstream sequences, there is a signal for addition of polyaethyl. So both the polyaethyl and the calf are usually important in maintaining the stability of the mRNA as well as the recognition of the synthesized messenger RNA. So this is actually the eukaryotic structure. We have regulatory region, we have regulatory sequence. This is the regulatory sequence, and then we have promoter sequence, and then we have 5' untranslated region. We also have 3' untranslated region, and of course we have exons and introns. So you see, these are our exons, and these are our introns. So usually these introns need to be spliced out because, of course, from the gene, it will now transcribe to produce messenger RNA, which is called primary transcript or immature messenger RNA. And from there, all the introns must be spliced out, must be removed out using what you call spliceosome complex. Now, all the introns should be removed, and then you left your sequence with only exons, which is the coding region, which will be used in the subsequent process, that is the translation of protein synthesis. So this is actually, so you see, in the case of eukaryotic gene structure, you see that it is more complicated compared to that of the prokaryotic system. So these are what are the exons and introns. So exons are the coding sequence transcribed and translated, and they are responsible for coding for amino acid in the polypeptide chain, and it varies in number. It varies in number, in sequence, and length. So a gene starts and ends with exons from 5' to 3'. Some exons include untranslated region because, the untranslated region also have some regulatory activity because we have what they call riboswitch. Riboswitches are also found in the untranslated region, and it's very, very important in terms of transcription, in terms of gene expression. And then the next thing is introns. So coding sequences are separated by non-coding sequences. So these non-coding sequences are called introns, and they are removed when the primary transcript is processed to give a mature mRNA. Because the primary transcript, you cannot use the primary transcript in the protein synthesis. Rather, you will have to use mature messenger RNA so that at least all the introns are removed by then. So all introns share the base sequences GT in the 5' direction and AG in the 3' direction. That is guanosine and thymine, and then adenine and guanine in the 3' end. So introns were first discovered in 1977 independently by Philip Sharp and Richard Roberts. So what are the eukaryotic structures, eukaryotic gene structure? So the significance of introns. So introns don't specify the synthesis of proteins, but have other important cellular activities. So they don't actually specify the types of the proteins that will be synthesized by messenger RNA, but it has some important cellular activities. So many introns encode RNAs that are major regulators of gene expression. So the introns that we are talking about is not that they are not producing some messenger RNA. Of course, they are producing some messenger RNA some RNA. An example of the RNA produced by the introns, it includes small nuclear, or maybe let me just say microRNA, and also small interfere RNA. So all these RNA, they are involved in gene regulation. That is like in a simple way, when we say microRNA and small interference RNA, they are just RNA interference. So what they do, this RNA interference, they regulate gene expression. They can also silence genes. They can also be used in knowing or tracing the gene function. So you can, in fact, you can even use them as a therapeutics. So many RNAs encode different types of, many introns are involved or are encoding different types of other RNAs that are important in regulating gene expression. And the examples, as I said, we have microRNA, we have small interfering RNA. So microRNA and small interfering RNA are just RNA interference. And RNA interference, we can use them in gene expression analysis. You can use them in gene function. In fact, it can even regulate the expression of particular proteins, that if there is a particular protein and it's produced in high concentration, then you can reduce the expression of that proteins within these regulators. You can also even increase the activities of some proteins by actually trying to maybe lower down the concentration of this RNA interference. And it also contains regulatory sequences that control transcription and mRNA processing. And also these introns allow exons to be joined in different combinations. That's alternative splicing. So in alternative splicing, you can remove different exons, and also you can make a different combination of the exons. And this results in the synthesis of different proteins from the same gene. So from the same gene, we can produce different proteins due to the alternative splicing. And also important role in evolution by facilitating the recombination between exons of different genes. So this exons is important in facilitating genetic recombination, which is actually involved in evolution. And this is what we call exon shuffling. So exons is important in exon shuffling. Yeah. And then the next thing is eukaryotic gene structure. In eukaryotic gene structure, as I said, it is more complicated than prokaryotic gene structure. So also in eukaryotic gene, we also have promoters. And these promoters is a regulatory region of DNA located upstream controlling gene expression. So that is what we call co-promoter. So a co-promoter, the transcription site is started at -34, which is the binding site for RNA polymerase, and it is a general transcription factor binding site. So that's a co-promoter, which is the RNA polymerase binding site, and it is -40, -34. And it is also a region where the transcription factors combine to initiate the transcription. There is also proximal promoter, which contain primary regulatory elements. So these together are responsible for binding up RNA polymerase 2, which is responsible for transcription. Then we also have the upstream 5'. So in the upstream 5', it have several functions, including mRNA transport and initiation of translation. Of course, when mRNA is being synthesized in the nucleus, it has to be transported from the nucleus to the cytoplasm, where there is a ribosome for the protein synthesis to start taking place. So this 5' untranslated region is important in transporting messenger RNA, and it's also important to initiate translation of proteins. That is the conversion of the matured mRNA into proteins. So the upstream 5' end is also involved in doing that. And there is also a signal for addition of 5' cap. So there is at the 5' end, there is addition of 5 cap, or there is addition of cap. And that 5 is 7-methyl guanosine, which is added to the 5' end of the messenger RNA. And this curve also facilitates the initiation of translation as well as stabilization of messenger RNA. So the 5' curve, which is added to the upstream region, it has three major functions. And number one is used to signal the ribosomes, that is a messenger RNA transported from the nucleus. So ribosomes recognize matured messenger RNA using the 5' cap. So 5' cap is used in recognition, and it also helps in initiation of translation, and also is important in stabilization of messenger RNA. And then the next thing we have downstream, which is the 3' end, and of course that's where we have 3' untranslated region, and it's served to add mRNA, and it's also bring the stability and attachment site for polyethyl. Polyethyl is also important in stabilizing messenger RNA, and the translation termination codon is also there at the 3' end, which is TAA, and then there's TAA, yeah, and then there's also AATAA sequence, which is signals also for addition of polyethyl. So these are the sequences that signal the polyadenylate polymerase to add polyethyl. And then we have terminator region. The terminator region is recognized by RNA polymerase as a signal to stop transcription, and we have enhancers, which enhances transcription of a gene up to a few thousand base pair offspring. And then there is also silencer, which reduce or shut down the expression of a nearby gene, that is silencer. So ladies and gentlemen, these are the silence features of genes. Number 1, the number of genes in each organism is more than the number of chromosomes. Hence, several genes are located on each chromosome. And then genes are arranged in a single linear order like beats on a string. Each gene occupies a specific position called locus. So there is a point or position where the genes are found, and that is what we call locus. And if the position of a gene change, if the position of a gene is changed, then the character will also change. And genes can be transmitted from parent to offspring. So if your parents are brilliant, the gene responsible for intelligence will be passed down to the offspring. So genes can be transmitted. The genes for beauty, the color of skin, can also be transmitted from parent to the offspring. Genes may exist in several alternative forms called alleles. Genes are capable of combined together or can be replicated during a cellular division. Genes may undergo for sudden changes in position and composition, and this is called mutation. So genes are capable of self-duplicating, producing their own exact copies. So these are some of the silence features of gene. These are the reference. Ladies and gentlemen, thank you for listening. Please, if you watch this video, subscribe and share.